Mitochondrial respiratory chain deficiency inhibits lysosomal hydrolysis. Mitochondria are key organelles for cellular metabolism and regulate several processes including cell death and macroautophagy. Here, we show that mitochondrial respiratory chain (RC) deficiency deactivates AMP-activated protein kinase (AMPK), a key regulator of energy homeostasis, signaling in tissue and in cultured cells. The deactivation of AMPK in RC deficiency is due to increased expression of the AMPK-inhibiting protein FLCN, folliculin. AMPK is found to be necessary for basal lysosomal function, and AMPK deactivation in RC deficiency inhibits lysosomal function by decreasing the activity of the lysosomal Ca2+ channel MCOLN1, mucolipin-1. MCOLN1 is regulated by phosphoinositide kinase PIK5 and its product pTDNs 3, 5, P2, which is also decreased in RC deficiency. Notably, reactivation of AMPK, in a PIK5-dependent manner, or of MCOLN1 in RC-deficient cells, restores lysosomal hydrolytic capacity. Building on these data and the literature, we propose that downregulation of the AMPK-PIK5 pTDNs 3, 5, P2, MCOLN1 pathway causes lysosomal Ca2+ accumulation and impaired lysosomal catabolism. Besides unveiling a novel role of AMPK in lysosomal function, this study points to the mechanism that links mitochondrial malfunction to impaired lysosomal catabolism, underscoring the importance of AMPK and the complexity of organelle cross-talk in the regulation of cellular homeostasis. Abbreviation: Mitochondrial transmembrane potential, AMP, adenosine monophosphate, AMPK, AMP-activated protein kinase, 8TG5, autophagy-related 5, ADP, adenosine triphosphate, ADP6V0A1, ADPase, H+ transporting, lysosomal, V0 subunit A1, ADP6V1A, ADPase, H+ transporting, lysosomal, V0 subunit A, BSA, bovine serum albumin, CCCP, carbonyl cyanide M-chlorophenylhydrazone, CREB1, CAMP response element binding protein 1, CTSD, cathepsin D, CTSF, cathepsin F, DMEM, Delbecco's modified Eagles medium, DMSO, dimethyl sulfoxide, EBSS, Earl's balanced salt solution, ER, endoplasmic reticulum, FBS, fetal bovine serum, FCCP, carbonyl cyanide P trifluoromethoxyphenylhydrazone, GFP, green fluorescent protein, GPN, glycerol-phenylalanine 2-naphthylamide, LAMP1, lysosomal associated membrane protein 1, MAP1-LC3B-LC3B, microtubule associated protein 1 light chain 3 beta, MCOLN1/TRPML1, mucolipin-1, MEF, mouse embryonic fibroblast, MITF, melanocyte-inducing transcription factor, ML1 and 2 GFP. Probe used to detect pTDNs 3, 5, P2 based on the transmembrane domain of MCOLN1, MTORC1, mechanistic target of rapamycin kinase complex 1, NDUFS4, NADH, ubiquinone-oxidoreductase subunit S4, OCR, oxygen consumption rate, PBS, phosphate buffered selene, pcDNA, plasmid cytomegalovirus promoter DNA, PCR, polymerase chain reaction, pTDNs 3P, phosphatidylinositol 3-phosphate, pTDNs 3, 5, P2, phosphatidylinositol 3, 5-bisphosphate, PIK5, phosphoinositide kinase, 5-type zinc finger-containing, PS, penicillin streptomycin, PVDF, polyvinylidene fluoride, qPCR, quantitative real-time polymerase chain reaction, RFP, red fluorescent protein, RNA, ribonucleic acid, SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis, shRNA, short hairpin RNA, cRNA, small interfering RNA, TFEB, transcription factor B, TFE3, transcription factor binding to IgHM enhancer 3, TMRM, tetramethylrhodamine, methyl ester, perchlorate, ULK1, UNC51 like autophagy activating kinase 1, ULK2, UNC51 like autophagy activating kinase 2, UQCRC1, ubiquinol cytochrome C reductase core protein 1, VAT-POS-A, vacuolar type H plus translocating at POS-A, WT, wild type.